All right, hello. So I'm going to be doing the pretest for biology version B. I am the case with no AI, so I will be studying only using Google. So I have a 20-minute timer here. I'm going to get it started right about now. All right, let's get right into it. This reminds me of a eukaryotic cell. Looks like a plant with like the rigid walls structure. Label 6. Um, Label 6, okay. That reminds me of the chloroplast because it's green and it has those stack structures. Label 4 represents which is necessary for the vacuole. That's also true, but this is their main function, I believe. Maintaining turgidity would be due to the cell wall, I believe. The cell membrane. The main function of the smooth ER represented by is, so you want the one with no ribosomes. Huh. I believe it might be number 2, but I still see ribosomes on it. It's just this is the only one I see with let me read the question again. Now, what I'm thinking is a smooth ER, if I remember correctly, might be for lipids, and transporting those vesicles. So I think it's number 2, and I'll go with lipid synthesis. I believe protein synthesis is the job of like the other organelles, such as like the nucleus, and then the ribosomes. So, yeah. Label 5 is present in, looks like that might be the cell wall. Plant cells only. The organelle labeled will be in higher numbers in muscle cells compared to fat cells. Probably mitochondria. Yeah, number 1. Because they need to burn more energy, I believe. But let's check the other ones. 5, 8, 10. 5, no. 8, less than nucleus, but not necessarily higher numbers. And 10, yeah, the same amount. So I think that's correct. The backbone polymer. Oh, I think it's rRNA. Yeah, RNA. Yeah, because it's a polymer and R stands for ribose rather than deoxyribose. So this is incorrect because it would use deoxyribose. I'm not sure, but I think it might be phosphate group, but it definitely has a nitrogenous base. if I remember correctly. Yeah. It could be this. Could also be this. These two are definitely untrue. Maybe this. Because you cannot do or yeah, I'm not. Yeah, I'm pretty sure it's this one. To be unzipped, replicated, and rezipped to function normally. Furthermore, the two strands may be necessary for proper folding. Because they might need like certain charges in certain spots for it to fold properly into a protein. I think that second part was a stronger answer. So I'm assuming this is a backbone of RNA, and then these are nitrogenous bases. Adenine, thymine. Yeah, I'm not really sure. I'm not sure what these are, so I'd have to go with this. I don't recall what these are. Okay, so adenine, thymine, guanine. Oh, geez, yeah. I'm really not sure. I've never really seen a NTP before. If I went with RNA for this question, I'll just stick with it, but I'm not too sure on these two. What is paradoxical? Huh. I'm not really sure. I'll just leave a dot so it doesn't block my answer, but I'll come back to this one. In this question, according to you answering. I'm assuming if it's RNA, that would come next, of course, but I'm not really sure. Isn't this a repeat question? Unless this was question 11 and this was question 12. I'm not sure. Now it's telling me a deoxyribose sugar is found in its backbone. But I believe this is a single strand. Yeah, it's confusing. This makes no sense. It's like contradicting itself. I thought it was an RNA, but it's telling me it's a DNA. But yeah, this is really confusing. Yeah, this is really confusing. I think I'm getting these wrong. Oh, maybe this is a little explanation. So this might be A and T. So this should be right because it's just according to the strandedness. Oh, and this is just their labels. Okay, it's making more sense now. Maybe the order of the maybe that's just a guess because I'm not sure. Maybe these two have to be switched. If it's thriving, then I think this might be the answer. I'm just trying to remember. Transcription is DNA to RNA, and well, if I'm going in reverse of that, it would be RNA to DNA. But I thought that was translation. I'm not really sure. I forgot all of this. I believe this might be right though. This might be the tertiary structure. Or no, primary is the chain, secondary is like the folding. I forgot what the third one was. Maybe spiraling. And then the fourth one is when like many of those spirals come together. So this is not true. This might be true. I'm not sure. I don't think the spirals is secondary, so I'll just go with this. It might be the strongest one. All right. Oh, I think I messed up on one of these. Yeah, it's not A and T. AUTA. I think it's this. Oh, but this is confusing. Yeah, I don't know about this question. But I don't think it's A and T. So you just have to G with C, T with U, T with A with U. So this would be C A U C A U. So it leaves these two. A C. I think that's right. What is most likely? What is the most likely reason that the sequence in the question has been written in reverse order? Is I don't really remember this. Oh, that's gonna take a long time. CAU A C A. time. Okay, I will just say that I didn't get this question, and whatever I get for these, I just omit it. Or I may as well try it, right? I don't know. Whatever, let's just submit. Okay. That's correct. Okay, I guess I thought that would be the cell wall, but I guess I was wrong. Oh yeah, because turgidity is where you like the vacuole pushes against the cell wall for it to be rigid. Oh, interesting. 9 lipid synthesis. Oh yeah, there's no ribosomes on that. Okay, that makes sense. I didn't know that was true. Okay, that's also true. RNA has one more hydroxyl group than DNA. I didn't know that. Oh, dude. I'm so. Yeah, I didn't realize it's thymine right there. And now that makes sense, right? Okay. I wasn't really sure about these. I think the ones I learned were named differently, so I didn't really want to perceive an answer for these, but I ran out of time anyways. Okay, so let's see what's next. Okay, I will come back in about 5 minutes. All right, I'm back. So I'm going to start my timer for 30 minutes right now. And we will begin the cell study. So I'm instructed to just search these on Google for 15 minutes. And at the end of the search, to append -AI. So I'm not entirely sure if I'm allowed to use this, but I will briefly read it anyways. So just around a regular shape, lacking a cell wall, that's a crucial detail, and containing various organelles. I'll have a look at maybe this one looks like a good photo. I'll have a look at this image. All right, nucleolus, nucleus, rough ER. Where's the smooth ER? Smooth ER is there. That's the question I got wrong. Centrioles. Oh, those are like the stacked, like long. I believe like in the diagrams I'm used to, they were like shaped like a star. I forgot what they were for, so maybe I'll give that a. Are they for storing something? Play a role in cell division and organization. Oh, okay. I think that rings a bell. Golgi apparatus, ribosomes. I'm pretty familiar with what most of these do. The vacuole is much smaller in this case. Microtubules. I'm not sure what those do. Oh, I think these just move things around. They're used in the one of the phases, like prophase, metaphase. Peroxisome, I'm not sure what that is. Yeah, I think this is one of like the less common ones that they test for. But cool, it looks like it's just for like digestion and detox. Mitochondria, I know. Lysosome, those are for breaking down and storing, I believe. Okay, so I think that was a helpful refresher. Maybe we'll look at one website here. Oh no, this is just art, I believe. Maybe this is a more helpful breakdown. Right, regulates passage. Cytoplasm, right, nucleus, DNA, nucleolus, ribosome synthesis. That's a good, helpful refresher. Mitochondria, ER. Okay, right. I think that's a helpful summary there. So let's try the next one. So let's try this website. Okay, this is interesting. Oh, this might be AI, so I'm not really sure. Yeah, it seems like a lot of these websites are not really helpful. They're just images of plant cells, which is definitely inconvenient for helping me study because it's taking some time for me to find. Like, I can't even find a website that'll give me like a description, really. But then again, I did Google for a diagram, but like, I was kind of hoping for like a diagram with like descriptions of what each one does, sort of like what's provided up here. Yeah, this is not too helpful. I'll refrain from glancing at this because this is AI. And yeah, perhaps let's just move on. I actually maybe I'll just quickly look at an image as I did previously. Yeah, same business. Peroxisome. Oh, I guess like peroxide is like a detox keyword there. That's a helpful memory aid. Not sure what this is. Maybe it's not that important for this test. Nuclear pore. That's where the DNA comes out. Smooth ER, rough ER, ribosome, chloroplast. Okay, yeah, it's all the um, basic structures I'm familiar with. Let's go ahead and try this. Okay, and yeah, with these websites, I find that they're quite annoying because there's a lot of text here, which will take a long time for me to read, and a lot of it might be unnecessary. You know, like sending impulses. I guess that's just to like set the scene for like how important they are, but like it's using kind of very beginner jargon. Cool gadgets, special air filter. Here's like a helpful table, I'd say. Sorry. Okay, so it gives a little analogy, which is helpful for a beginner. Yeah, and this is a concise function summary, which is nice. A very simplistic image here, maybe perhaps too simplistic for like the case of studying, so this might not be a helpful diagram. And yeah, it's going into a lot of detail. This will take far too long for me to read. And I am fairly confident I'm familiar with most of the primary cell organelles and structures. Okay, this should be 5 key differences, should be a helpful brief comparison. Okay, great. Adenine. Oh, right, so the order was switched. Okay, that's a helpful refresher, because in the first test I noticed that it was A and then T, and then it would be like or yeah, I actually don't remember it that well, but yeah, read time 7 minutes, and I think that might be a little too long. DNA, RNA, DNA replicates, right? I would like to know what was the difference between 3' and 5' RNA. Oh yeah, because the like the sugar has, I think it was either like 5 or 6, and it's like which one it attaches to. So why would the direction of the 5' and 3' switch for RNA? Okay, I think that makes sense. This is a helpful bit of refresher. Okay, so it's a more stable molecule. That was one of the questions I missed on the previous test. Interesting. I didn't know about this. Okay, thanks. I'm thankful that they displayed this, because I was a little rusty on this. Transcriptions, transports to the ribosome, cellular factories, tRNA. Okay, I remember that. rRNA. Okay, yeah, I remember all of that. Okay, where's the 2 hydroxyl groups? So there's one here, I'm noticing. And that might be it. Okay, I think I got all of the information from this website I needed. This was actually rather helpful, I'd say. The thing I liked about it was it provided information that I didn't really think of, but was really helpful to know, clearing up a lot of test questions I had. For instance, like it prepared me with like the mRNA, tRNA, because I was rusty on this. I like this table. It shows some special information that was important, and yeah, this was concise and helpful. Did I? So this seems like it's giving me the same output. And I believe I already got a little. Yeah, okay. So that website summed it up for me. I'll read this little paragraph here. Yeah, I think that was also. It wasn't like it didn't really provide me any new information, I believe. I guess this was helpful. So I've got about 1 minute left. So let's try this one. So I'm looking for a quick. Okay, that gave me a sense of what it was. I'd say it did a fair job with that. And let's try this one. I usually like these drop downs because they're quite brief. However, it can be like sometimes not exactly what I'm looking for. And it may teach me something that's not what I'm looking for. Okay, since I don't have much time, let's try this one actually. Okay, yes, so deletion, duplication, invert. Now, this wasn't the same, this was the vocabulary I was used to, but that wasn't what appeared in the test. So okay, this is exactly it. So I'm just gonna, I think this is important, so I'm gonna read this even if I go a little over time. Okay, I remember this. Okay, so this is not too bad. This is like harmless. This is a little worse, and this is the worst, I believe. Okay, that's fairly straightforward. So let's move on to the next section. So I believe that would be the nucleus where the DNA is held. The smooth produces lipids, and the rough produces it's not ribosomes. It's oh yeah, I actually forgot. I'm gonna go with proteins, but I don't think that's exactly true because the nucleolus produces ribosomes. The ribosomes go to the rough ER, and it might be vesicles, but I'm not really sure. I'd just like to check my answer for this one. Which organelle is necessary for cell division? I'm not really sure. I'm thinking maybe the cell membrane because that's what is flexible, and maybe it's just referring to like the DNA. I think this one's a bit vague, so I'm not really sure about this one. It could be like the nucleus. because you need DNA. Actually, let's just go with that. Choose a symbol on the keyboard that best corresponds to DNA. Maybe an equal sign. I'm not sure. I'm looking at my keyboard now, and I don't think I see any other. Or an X, maybe like a chromosome, because it's like a spiraled helix. It could be X or an equal sign. I'm not sure, but it does say symbol, not letter. Okay, let's go with equal sign. And this would be a hyphen. Yeah, I think that makes sense. For the flow of information in a cell. Oh, it says in one direction, so I would assume this. I know that this is like the correct flow. I'm not sure what P and A is, so I guess it's just got to be this one. Oh, that is false. It's stored in the nucleus. A pairs with T. Nothing. The two main types of secondary structure are alpha. I'm not too sure, but I do know that these are two words that ring a bell. I know it's likely not spirals and sheets. I don't think it's fingers. Yeah, I think I'm gonna go with this one. So we have some time left. It looks all right. So I'm gonna submit this and check my answers after. That way I can get some feedback right away. Okay. Okay, centriole. Okay, I believe that's the one that pulls the things apart, right? Yeah. Essential for cell division. Okay, that's fair. I did not remember that. Helices and sheets. So let's try this question. Right, hydrogen bonding. Backbone atoms. Okay, so maybe it would be primary, secondary, tertiary structure of protein. Oh, and quaternary is when they okay, yeah, this is helpful. I fully remember this now. Any other questions? I believe that was all of them. So I still have some extra time. So let's go ahead and Google some of the answers I got wrong on this one. I know why this one's wrong. I know why that one's wrong. So I believe that that was referring to the cell membrane, and I thought it was the cell wall. So okay, I I just want to clarify this. Okay, so it might not be the cell membrane. Then what is it referring to? Or maybe it is, but the word "some" is a little maybe not fully correct. I think it has to be all of them. This makes sense. Okay. So it's saying that this must be A and this is T, but how is that possible? So this is T A A T. That's strange. Yeah, I don't really understand this question. So let's quickly do this one. CAU, CAU, histamine, AAC. I don't know what ASN is. UU. CAC, histamine. Yeah, that's what I think I got. Yeah, okay. So I actually already forgot which one corresponds to which. So this is nothing happens, just a one error. So different amino acid. So these two basically are the same. It's just this one's more significant just by chance. And this one just ends early. Okay. Okay, so I think, yeah, silent. Yep. Okay, and I'm going to do this one without looking. One point mutation occurs. The second one interacts with the first one. Maybe this one. Nonsense. Okay. Yeah, okay. I guess I thought that this one would mean that it created a stop codon. But I guess this one just means that it's a separate polypeptide. Or sorry, separate yeah. Okay, I'm not really familiar with this terminology. So silent is the same one created, so nothing happens. missense is a mutation where a different amino acid is used, and nonsense is the worst one, where it's like either stopped early or creating like a protein that's not folding properly. But overall, though, like I think I am familiar with most of it now. Let's see if there's any other questions. Maybe let's look at this a bit more. Yeah, it's kind of hard to find like a relevant website. This process is a little time consuming, and this website's taking a long time to load. Okay, well, I have 1 minute left, so I think I'll just move on. Take a 10-minute break. Okay, so yeah, I'll come back in 10 minutes to do the post test. All right, I'm back. So now I'm going to do the post test version A. So I'm going to set up a 20-minute timer. Okay, it started, and let's get right into it. So this is the centrioles, and yeah, I just Googled that. I believe it is the cytoskeleton. Yep, protection, maintenance of cell shape. Yep, that seems right. So I do know that they are the centrioles, and I believe cell division is correct. I believe they pull apart the DNA. That was something I did Google for. So that was actually like a helpful thing to search for, the organelle. Those look like ribosomes because they're here in all of this. And so rough endo. Nucleus. Well, they're produced in the nucleolus. Yeah, I'm not really sure. It could also be like cytoplasm because they just float around. Maybe that might be a stronger answer. Because they're not really found inside the nucleus. They're found like kind of right there outside. They're produced in the nucleolus. Oh, the Golgi apparatus. I believe this is for folding proteins. And it looks like an ER, yeah, so it's K, I believe. And yes, that's correct. Oh, it's for like packaging them, I believe. That looks like the nucleolus. Oh, jeez. I don't recall exactly, but I believe it should be that because it's responsible for ribosomal ribosome production, and cells need ribosomes. So I would assume that it must be present in every single type of cell. Oh, this looks like the same question as last time. I believe that's H, the mitochondria. Mitochondrion. Okay, let's move on. Nucleotide. It's pretty self-explanatory, I think. I believe it's a sulfate group, which is incorrect. I think it's a phosphate. I believe that should be true. I know it is definitely more stable. I'm a little unsure about reactivity, but I would assume that if it's more stable, then in turn it would be less reactive. Because those are like stable. Being stable means to be less reactive. DNA lacks the hydroxide, which is present in RNA. Therefore, it is much more stable and hence less reactive. That should be sufficient. Okay, now this is much easier to read, I think. It's definitely a DNA. And because there's U present, this has got to be RNA. The double-stranded nature of the diagram must mean that it is DNA. However, excuse me, the uracil base means it must also be RNA at the same time, contributing to this paradox. So we'd be looking at these. So this must be A, and this must be U. I'm assuming that that's just a repeated question again. I forgot which carbon's number one. Is it 1, 2, 3, 4, 5? No, it's 1, 2, 3, 4, 5. Or 1, 2, 3, 4, 5. Yep, so there's a hydroxyl to this carbon 5, carbon 2. Or no, 2 and 3. Well, I'm just assuming that since there must be an extra hydroxyl from like the images I saw, this must be RNA, but I kind of forgot how to read like the order. This is so it is RNA. guess we could say that. Okay. Okay, this is the I forgot what it was called. But I googled it. Protein. Transcription. Translation. That's incorrect. This might be correct. That's not correct. Yeah, I believe it's this one. Okay, so GC, CUC. So it's between this. No, it must be this one. Right? Yeah. CUC. This is the only one with CUC. Right? So if it's reversed, then we must do GAC. GAC. Aspartame. UCA. UCA. AUA. AUA. UUG. Leucine. CUC. Again, okay. I believe that's the only relevant one. This would be a missense. Okay, I'm fairly confident in all my answers. I'm going to quickly double check it because I have a lot of time. This should be correct. That should be correct. I'm a little unsure about this one. So D, these are definitely ribosomes, and they're present in the smooth ER, or rough ER rather, and they're produced in the nucleolus, but they're found in the cytoplasm. Yeah, let's stick with the cytoplasm. Golgi apparatus, sending and receiving, yes. Labeled C. Nucleolus, it must be present in both because of ribosome production. Yes, okay. Fairly confident of these responses. Yes. I think it's a phosphate group. That's self-explanatory, and I was confident in the description. Explanation, sorry. DNA, RNA. Yep. AU. AU. Yep. Oh, this is already 5-3. And then this is 3-5. So I think this is actually backwards. So it's Lou. So let's actually check this. So it's trying to trick me. I think this means that it's written the same way as this, which means I must read it like this. Okay, that was a clever little trick. I hope I did it right. Let's just see. So CUC is leucine, UG, leucine. Yeah, it should be correct. AUA. Yep. UCA. Yep, and then JAC. Yep. Okay. Glad I caught that. That seems correct. Yep, because I believe if you change it to a stop codon, but the rest of the oh, no, I think that's correct. Yeah, I think I'm just overthinking it. So this is definitely right. If we stop it early, it's definitely not going to be the correct protein. Yeah, I think this is the only one that makes sense. I'm just going to thoroughly read this question. Seems correct. Okay, I believe I'm done. So I'm going to submit it and check my answers. Okay, beautiful. That's the first answer. Yeah. So yeah, okay, that's the same response. T. Huh? Oh. Oh, because if this is DNA, then it should be a T. Oh, okay. So I feel like if there's question numbers, I would have. Okay, I get it now. They're saying that if this were DNA, then this must be T. But I just felt that since it was RNA because of this U, then it was just a repeat question. But okay, that makes sense. That's correct. Oh, interesting. Whoa, whoa, whoa. That's totally different from what I had. How can this be right? Interesting. I'm not sure about that. Okay, well, it appears that I got this one wrong for some reason and this one wrong. So that's 2 and this one wrong. So it's not too bad. I think 3 wrong is fair. So my comments about using Google are, I would say it's much more time consuming to find the information you're actually looking for, and a lot of the information you may read will be irrelevant. So it is definitely frustrating and, of course, much more time consuming. As a student, if I'm in a hurry to study, I may be very impatient and not wanting to read all of this unnecessary wording, and I may miss what I'm actually looking for. Furthermore, finding like actual good sources such as this one may not always come along, and I do like these, although sometimes they may be a little bit off topic. Yeah, overall, I think it was definitely an improvement. I think a lot of the improvement just came from being exposed to this test previously, like a similar version of it. So that helped me gauge my time better, and I was more familiar with like what to look for. I didn't have to read the questions for as long, and yeah. So those are my comments. Thank you very much.